Grüezi ragazzi, willkommen e bienvenue to Cannabiscienza's first LinkedIn Live of 2023. We have a lot of very interesting conversations planned this year, and today is no different. To those of you joining us live, thank you very much for being here with us right now. And for those that will be checking out the recording at a later point in time, thank you very much for checking us out. I hope that you guys enjoy the session and that you may have learned something today. A little quick intro about Cannabiscienza. We as a team aim to combat the bottlenecks for the medical cannabis industry, and we do this using the power of education and through the training of healthcare professionals on the effective use of cannabinoid medicines for a number of different pathologies. In doing so, we believe that we are accomplishing also our mission of increasing access to medical cannabis for patients in a number of different countries. And if you haven't already, head over to our LinkedIn page, give us a follow, check out our website, and stay tuned for the upcoming events that we have planned. Joining me on today's call is Fabio Turco, who is the CSO for Cannabiscienza, and I will let Fabio introduce our guest today. Thank you.
Yes, Bijan, thank you for the introduction. Good afternoon to everyone. Welcome to today's live, and which topic is the role of the CB1 receptor in human pathophysiology. It's a great pleasure to have with us Luigi Bellocchio, researcher and associate faculty member at the Neurocenter Megandi in Bordeaux, France, who is author of very relevant publication on important journals about the endocannabinoid system, especially regarding the role of the cannabinoid receptor 1, simply known as CB1 receptor, which will be the main character of today's topic. Just to introduce the topic, we know that the CB1 receptor was discovered in the 1980s when it was clear that following the discovery of the THC by Meshulam and co-worker, that should be an endogenous receptor responsible for the effect of THC and cannabis. So, thanks to the work of Arlene Howlett and his lab at St. Louis University in the USA, the CB1 receptor was discovered and later cloned. This was the first step that led to the discovery of what was subsequently called and named the endocannabinoid system. Now we know that the CB1 receptor are metabotropic receptor with the 7 transmembrane domains coupled with to a G inhibitory proteins, which decrease the intracellular concentration of the cyclic AMP. This exerts an overall inhibitory effect on cellular activity. We also know that CB1 binds THC and is thought to produce the psychotropic effect of cannabis. But let's start from the physiology. So I want to give the talk to Luigi Bellocchio. And my first question for this is where the CB1 receptor are located in the central nervous system, and how does the CB1 receptor affect the brain physiology?
So CB1 receptor is almost everywhere in the brain, and we can define it as the most abundant G protein coupled receptor expressed in the human brain and the mammalian brain in general, but it's also in peripheral nervous system, and this is something that, if you want, we can talk about it later when we afford some other question. And so CB1 but it's quite heterogeneous because the expression of the receptor, it depends really on cell type, on the brain region, and also on the pathophysiological state involved. We can say that the brain physiology, it's almost all affected and modulated by the action of CB1, and there are more and more discovery of several physiological processes that are regulated by CB1. Let's say that is a system that keeps homeostasis of the body and especially of the central nervous system. So it has to be in a certain state while going lower or higher than its physiological activation. And this will come soon when we'll talk also about the effect of THC. When the system goes out, we go in pathological ranges and pathological situations.
Okay. As I was looking at your bibliography or PubMed, and a title caught my attention. It was Cannabinoid Receptor Type 1, Breaking the Dogma. So my question is, what are the dogmas that must be deconstructed or broken down regarding the CB1 receptor?
Well, there are several dogmas, actually. The first one is the cell type, because CB1 receptor, actually, if you look at the gene name, stands for CNR1, which means cannabinoid neuronal receptor 1. So this is already one dogma that we and several other groups contributed to break, which is not only neuronal receptor, but it's also in the brain, it's also present in astrocytes, in microglia, in oligodendrocytes. And this is thanks to the mouse genetic that allows selective deletion of several receptor pools. Well, also in the neurons, CB1, another dogma is that CB1 is mostly on GABAergic neurons, which has the high effect, with a huge negligence on the field on CB1 receptor on glutamatergic neurons, which actually it's very few, but it has key roles. For example, the anti-epileptic role of cannabinoids, the appetite promoting, the effect of fear extinction, they all rely on the activation of this very small pool of CB1 receptor, which actually, if you knock it out and you do studies of cannabinoid signaling in the brain, you actually found that the majority is mediated by this small pool of glutamatergic CB1 receptor. This is another dogma. And I should say, so the astrocytic, the GABAergic versus glutamatergic, which also make the dogma that quality, it's also actually, has always been taught to be dependent on the quantity, which is not the case for CB1, because you have very small pool, almost undetectable, but they play key roles in the brain physiology and also in pathology. And of course, another dogma is the subcellular localization of receptor, because people used to think, okay, it's a G protein coupled receptor, it's on the plasma membrane, but this is not actually true, because CB1 is also present in some intracellular compartments, such as endosomes, and more importantly, for what we have discovered in our lab, and other people are still further confirming this observation, it's present on the mitochondrial membranes.
Okay, and so we have this different intracellular localization of the CB1 receptor. But how it affects the different action that the receptor can accept, and is this relevant to therapy with cannabinoids?
Well, CB1 receptor on mitochondria, it actually has two biochemical readouts when activated. First is to inhibit mitochondrial activity, and so devoid neurons of ATP coming from mitochondrial respiration. But in the same time, in other cell types such as astrocytes, it's coupled to increase in calcium mitochondrial import, which helps to maintain the physiological calcium balance of the cells, especially in astrocytes, where calcium is the most important mediator for all the release of several molecules that helps neurons, and also interferes between, there is a concerted effect of respiratory action and calcium and mitochondrial calcium also on the lactate release, which is the process through which astrocytes feed neurons. I should say that for therapy, this is a further, so CB1 for therapy, it's in general, it's very important to modulate the action of CB1 because it's a buffer system for excessive neuronal activity. But another way can also be a buffer for excessive mitochondrial activity, where we know that an excessive mitochondrial activity is not always good because it results in less ATP and more reactive oxygen species, which are also buffered by the action of CB1. So we should say that it's a further level of control in the homeostasis, which not only goes at the circuit or cellular level, but also in intracellular processes.
So do you think it's possible to have some kind of compound that can selectively bind to mitochondria CB1 or to membrane CB1. Do you think it's possible to have this kind of compounds?
Well, so far we have been used, for example, to study mitochondrial CB1. We have been using some antagonists which are cell impermeable. So it means that they don't enter in the cells and they likely only block plasma membrane versus mitochondrial CB1. There is a group in Spain which is working for agonists which have the same principle. And we with the genetic manipulation in mice, we were able to selectively knock out, so remove the receptor only mitochondria, which is a good tool to study. But yes, this is the future for the study to really see when we identify some action of intracellular CB1 to try to block only the plasma membrane. Another good thing can be also to use this mitochondrial medicine, which is a field which is taking place. And it's based on the fact that you can add to some compounds some moieties that they charge them negatively and they target them to mitochondria. So also one idea can be to stimulate only mitochondria or inhibit only mitochondrial receptor, for example.
Okay, because I think that stimulating mitochondrial receptor exerts different action compared to membrane CB1 receptor, right?
Yes. So you have to think in proportion, because mitochondrial CB1 receptor, it's only 10% of the receptor. The majority is on plasma membrane, 10% is on mitochondria. So it's actually like a second hit. It means that when you have few agonists, which can be exogenous or endogenous, at least to our observation in preclinical models, they don't seem to hit mitochondrial CB1. But when you have loads of agonists or a huge stimulation that triggers a massive release of endogenous cannabinoids, because these endocannabinoids are released on demand, so they are not always present, this will hit mitochondrial CB1. And this can be a way also to explain why high doses of cannabinoids, they have some side effect like motor or memory impairment, which actually triggers mitochondrial CB1. Indeed, if we remove mitochondrial CB1 in mice, we still have the action of endogenous cannabinoids, so the system is not blocked, still functional, but we lose, for example, the side effect of high doses of exogenous agonists. And this for me, one of the key for the future of the therapeutical cannabis.
Yes, of course, because we know that one of the main problems regarding the use of cannabis are the psychotropic side effect that maybe when people use cannabis like a recreational drug, they are the effect that they want. But using medical cannabis, these are regarded as a side effect. So finding a way to separate therapeutic from side effect would be a huge aim for the research in the future. Well, let's go in another way. Let's go in the periphery, and because in the last years there is a huge amount of research that has demonstrated that the gut and the brain interact across the so-called gut-brain axis. Does this interaction involve the CB1 receptor too?
Yes, of course, because you have a CB1 in all levels. You have CB1 in intestinal epithelial cells. You have CB1 in vagal afferents going from the gut to the brain. And then you have also CB1 in noradrenergic afferents that normally goes from the brain to the gastrointestinal system. And to this regard, we actually performed a series of studies some years ago aimed at identifying through which circuits rimonabant, which is the selective blocker of CB1, has its appetite inhibitory effect. Because we know that rimonabant is a strong anorectic drug, which also promotes it for obesity. But this drug was discarded by the market because of the very important central side effects.
Yes.
However, what we found that actually the effect of rimonabant in feeding is peripheral. It's actually a blockade of CB1 in the gastrointestinal system, which will not buffer anymore an excessive noradrenaline release, this will, through the guts and through beta-adrenergic receptor, will stimulate vagal afferents, which will send satiety signal to the brain. So it's a peripheral satiety mechanism which is integrated into the brain, but the CB1 involved in this, and we found it with mutagenesis and local and wider blockade, it's actually happening in the periphery. So this gut-brain axis is, and can be in the future, one way to rethink about CB1 blockers for the therapy of obesity, for example.
Okay. And to your knowledge, are there any compounds that can block CB1 just in the periphery?
Well, here you touch a pain spot for me, because there are some papers, but and this drug, they really actually really work. So for the periphery, for the possible application in therapy, they are very good because they are devoid of side effects of total CB1 antagonists. The only problem of this drug in research is that they seem not to be specific for CB1. So if you want to use them in therapy, good. But if you want to use them in basic research to understand the role of peripheral CB1, they are not actually the best tools.
Okay, but maybe, so you say that they can be relevant to therapy, to anti-obesity therapy.
In clinical model, they work. They are as efficient as rimonabant, which is the full CB1 antagonist, but they don't have its side effect.
Yeah, this is a good news for therapy, I think. Another question. So several of your papers have been published in very prestigious journals such as Nature, Science, and Neuron. Is there a research project that you are particularly proud of?
Well, there are several. I should say that my two favorites is my paper I published in 2010 from my PhD, showing the bimodal control of food intake by the endocannabinoid system, because we were actually the first to show a base for the anorectic effect of high doses of cannabinoids, which is not sedation, but it's actually a decrease of the rewarding properties of the food for mice through GABAergic CB1 receptor, while glutamatergic CB1 receptor, which is a small amount of CB1, it's the classical anorectic orexigenic promoter by cannabis induction. And this is a mechanism that we also in another paper found to involve actually olfaction. And another paper I'm very proud of is the Neuron paper, the project that underlied the Neuron paper we published in 2001, where we found that there is one neuronal circuit that expresses a lot of CB1, which mediates both a good effect of cannabinoids, which is the analgesic one, and the bad one, which is the cataleptic. So the impairment of motor reflexes, which is normally the cause of car accident upon cannabis. And here we found actually that is the same receptor in the same neurons, but on two different cellular pools, which are mitochondria versus plasma membrane. And the plasma membrane CB1 seems to be the mediator of the analgesic properties through modulation of neuropeptide release, whereas mitochondrial CB1, it's actually the responsible for the cataleptic and the motor impairment effect of cannabinoids via the classical inhibition of neurotransmitter by reducing mitochondrial activity and ATP on the synapses. So these are the two projects I'm quite proud of.
These are very interesting projects. And I was thinking that, you are based in France, and situation in France about the medical cannabis is not so open. There are still a lot of taboo. So I was thinking, how was your work received by France people and France Academy?
Well, by France Academy, all the work in people are very interested in work in neuroscience, especially when you are proposing novel ideas. I should say that to my gut feeling is that the work we do, we use CB1 to understand how the brain works. And this is actually the best field and the best system to study the brain functioning, the endocannabinoid system, because it's always everywhere, and it allows you, through just one receptor, to identify several roles of intracellular compartments or different cellular population, etc. But the problem is that we have so far some huge problems in making press release and communication to grand public of our research, because you have always to be careful that not to say that your research are there for promoting medical cannabis, because, and also this is some problem when we ask for national fundings.
I think this is quite disappointing, because your research are considered in all the world as very good at innovating research. So maybe I don't want to say too much thing, but maybe government should take more attention about what you and your colleagues do.
Interviewing scientists now. They invite scientists. They ask us to write reports. So the situation it's anyway moving somehow.
Okay, maybe in the future things will change how they are changing in the world regarding medical cannabis.
They have a lot of interest here in France now on CBD.
Yes, yeah. I know that they changed the law. Now it's permitted. So maybe things, in all the Europe, we are going towards a more open view about medical cannabis, also recreational cannabis. So I think at a certain point, all the European countries must be aligned. Let's go on and with another personal question about what you think about the future of the research on the endocannabinoid system. Do you think that we still need to understand more about its pathophysiological role?
I think so, because there have been in the last year some discoveries that we would never have thought of, processing that are mediated by CB1, or some particular new role of CB1 which has been appeared in scientific literature. So I think this is a continuous evolving field, not only for the cannabis, for therapeutical cannabis, or for finding novel therapeutics which are based on it, but actually because, as I told you before, this is the best way to understand the functioning of the brain. Because it's not an activator or inhibitory system that now, with the optogenetics, chemogenetic, and all techniques for neural manipulation, we basically are discovering how the circuit works, because you activate a circuit, you have some physiological and behavioral readout, and this is okay. There are plenty of papers coming on now in top journals. But for what is behind the modulation, I think this is the key to still keep going on in research, and the endocannabinoid system, in particular CB1, is one of the best characterized, and one of, not although the best characterized, the one that is given more and more surprises.
May I ask a quick question? And I have to admit, as somebody that is not, doesn't have the scientific experience and background as you guys, it's a pleasure listening to you. But I have a very basic question, and perhaps silly, but what is the difference between, or the relationship between the CB1 and the CB2 receptor?
Well,
if short answer.
There is a big debate about CB2 receptor, because some people say it's in the brain, some people they contradict them soon. For sure, CB2 receptor in the brain is probably there. It's just that the technical expertise we have today does not allow us to see it in a specific way like CB1, but seems to be involved also in several processes mostly related to neuroinflammation, for example, on microglia. And for example, in Huntington disease, we know that CB1 receptor is protective, but CB2 as well. So the combination of two receptors is probably a way to keep the homeostasis.
Very interesting.
Yeah, maybe we need more research to find out the role of CB2 receptor in the brain, if they are in the brain, which is its role. Well, we are running to the end of this talk, so I have another question for you. Would you mind to tell us about the current research projects you are working on there in France, and what are your plans for your future?
So on the current research in which I'm working on, I have two big lines that I'm following. One is that I decided to leave the very small amount of CB1, and now I'm focusing a lot my attention on the striatonigral and striatopallidal CB1, which are the readout of the striatum, which is the key of the basal ganglia, and seems, for example, that striatopallidal CB1 seems to be very important for especially recreational effects of drug of abuse, such amphetamine and cocaine, for example, in the modulation of basal ganglia, and where there seems to be some new form of synaptic plasticity that we are going to discover. This is one project. Another one is a little bit more ambitious, but seems to be in the right way. It's just that we took advantage of the fact that CB1 modulates mitochondrial activity to actually invent a tool which will allow us to, in a chemogenetic way, to stimulate mitochondria. And this will, for us, be a new line of research to really link with
Luigi, I think we lost you for the last 3 seconds. This will be the research to link, and that's when it's
mitochondrial activity with behavior, to provide a functional link between mitochondrial activity and behavior.
There we go. Perfect. Thank you. Thank you. Sorry about that to our audience. There may have been some connectivity issues, but I mean, again, for me, it was an absolute pleasure listening in today, and I'm glad that Fabio asked that question just now about what you have going on and what's in the pipeline, because I think it only makes sense if we reconvene here again together down the line, and you can give us an update on what's been happening. And I guess also on this note, to anyone in the audience, we as a team are exploring also what we can do to help the development of the market in France. So that means also doing our part to educate the stakeholders of the industry, working with people like Luigi who really spend day in and day out working with the plant and doing this research. And we will be hopefully making some announcements soon about what we got going on in France. And if anyone's listening, if you need training, which Luigi, you have the view from the ground there, but keep an eye out for what we have going on in France, certainly, and we'll definitely have to have you back for another call to update us on what's going on.
Yes.
Lovely. Thank you, gentlemen. Thank you to everyone in the audience. Thank you to those who have commented. If you have any last-minute questions, please post them in the comments now. Don't forget to give us a like, and thank you, Luigi, for your time again today. Thank you to everybody that joined us or that will tune in later, and have a great rest of the evening.
Okay. Thank you, Luigi. Thank you, everyone, for attending us to this live, and see you next week with Cannabis Scienza live on LinkedIn.
That's right.
Goodbye.
Bye.
Ciao.